Hello students, today we will be doing a video on Histology of Integumentary System Part 1. Now the integumentary system consists of skin and its appendages. Skin made up of two layers, epidermis and dermis, along with hair follicles and hair with the pilosebaceous unit, sweat glands, nails and mammary glands. Hypodermis consists of adipose tissue that lies deep to the dermis. Hypodermis means subcutaneous fascia. Skin forms the outer covering of the body. It is the largest organ of the body, constituting about 15 to 20% of the total body weight. Skin has two layers, superficial epidermis and deep dermis. Let us see what are the types of skin. Based on the thickness of epidermis, skin is of two types. Thick skin or glabrous skin has a very thick layer of epidermis. It shows thick stratum corneum, which is the outermost layer, and it does not have hairs. And it is located in the skin of palm of the hand and sole of the foot. The other type is thin skin, wherein epidermis is thin and shows the presence of hairs, and it is located in the skin covering all parts of the body except that seen in the palm of hand and sole of foot. Let us now see structure of skin. Histologically, it consists of two layers, epidermis and dermis. Epidermis is a superficial layer that consists of keratinized stratified squamous epithelium. Dermis is a deep layer made up of connective tissue. The epidermis, as I said, consists of keratinized stratified squamous epithelium. It shows 5 layers of cells. From deep to superficial, these are Stratum basale, Stratum Spinosum, Stratum Granulosum, Stratum Lucidum, which is seen only in thick skin, and Stratum Corneum, which is the superficial most layer of skin. Stratum basale, also known as Stratum germinativum, deepest layer of epidermis, shows a single layer of cuboidal or low columnar cells resting on the basal lamina. Cells shows basophilic cytoplasm and melanin pigments. This here we are seeing a hand-drawn diagram of thick skin. What we see here is the epidermis. This here is the deepest layer resting on the basal lamina, and that is the stratum basale, made up of cuboidal or low columnar cells, all resting on the basal lamina. So that is the stratum basale, deepest layer of epidermis. Function of this layer is that the cells here divide mitotically to give rise to keratinocytes that form superficial layers of epidermis. Hence, stratum basale is also called as the stratum germinativum. Next we see the stratum spinosum, also known as the Malpighian or prickle cell layer. It is made up of several layers of keratinocytes, which are polygonal cells connected to adjacent cells by desmosomes. During tissue processing, cells often shrink, but these keratinocytes remain adherent with each other at the sites of desmosomes. This creates spines over the surface of these cells. which are seen at the site of the connection due to the desmosomes. Hence, they appear as spinous or prickle cells, and this is also called as the prickle cell layer. Few cells of stratum spinosum may undergo mitosis. Hence, stratum spinosum and stratum basale are together considered as the germinative zone. The deeper layers of cells are polygonal with spherical nucleus, while the superficial cells are flat. Same diagram, we see this stratum spinosum. Cells which are polygonal, having a spherical nucleus. As we go towards the superficial layers, the cells become flattened. So that is stratum spinosum or the prickle cell layer. Next we see the stratum granulosum. Consists of 4 to 5 cell thick layer of flattened cells called keratinocytes. These cells contain granules of keratohyalin protein, hence called as stratum granulosum. Keratohyalin is a precursor of filaggrin protein, which on aggregation forms the keratin filaments. On H&E staining, these cells appear intensely basophilic because of these granules. What we see here is the stratum granulosum, 4 to 5 cell layer thick, presence of flattened cells which are the keratinocytes, showing the keratohyalin granules that give the intense basophilic appearance. The next layer is the stratum lucidum, present only in thick skin. On H&E staining, cells are lightly eosinophilic and appear homogeneous. The cell boundaries are indistinct. Nuclei and cell organelles become disrupted and disappear towards the superficial layer. This is a transitional layer of cell death. This layer which we see here is the stratum lucidum, a homogeneous eosinophilic layer. We cannot appreciate the cell boundaries. No nuclei and cell organelles seen, as it's a transitional layer of cell death. Next we see the stratum corneum, most superficial layer of epidermis. It is 15 to 20 cell thick layer. Cells are non-nucleated, flat, scale-like, squamous, dead cells, and they do not have cell organelles. They are filled with keratin filaments. Cells are held together by extracellular lipids and carbohydrates that form a glue-like substance. This we see the superficial most layer of epidermis, which is the stratum corneum, showing only the cell outlines or the cell boundaries with no nuclei and no cell organelles. The extracellular lipids make skin water impermeable. Thickness of stratum corneum depends on the amount of friction it is exposed to. Hence, this layer is very thick in the palms and soles. The superficial cells of this stratum are constantly shed off and are replaced by proliferation of stratum germinativum. Next we see the dermis. Dermis is made up of connective tissue containing collagen bundles, blood vessels, lymphatics and nerve fibers. It has two layers, papillary layer and reticular layer. Papillary layer of dermis lies just beneath the basal lamina of epidermis It consists of loose connective tissue. It shows finger-like projections in the undersurface of epidermis that are known as the dermal papillae. Epidermis also protrudes into the dermis in between the adjacent dermal papillae, and that is what forms the epidermal ridges. This layer contains type 1 and type 3 collagen fibers. Blood vessels of papillary layer do not penetrate epidermis. Epidermis is avascular and receives nutrition by diffusion. Nerves from the papillary layer may terminate in the basal layer of epidermis. The same diagram here where we will see the parts of the dermis. This here is the dermis. This projection of dermis into the undersurface of epidermis is what is called as dermal papilla. In between adjacent dermal papillae, the part of epidermis which is seen is known as the epidermal ridge. Reticular layer of dermis lies deep to the papillary layer and is made up of dense irregular connective tissue. It is thicker and less cellular than the papillary layer. It consists of thick irregular bundles of type 1 collagen fibers and elastic fibers. This here, which we see lower down, is the reticular layer of dermis. So two layers of dermis, the papillary layer and the reticular layer. What we also see here is a sweat gland showing the duct and the secretory components. Now let us see what are the differences between the thick skin and the thin skin. Stratum lucidum is present in thick skin and is absent in thin skin. The thickness of epidermis is between 0.6 to 4.5 millimeters in thick skin, and it is between 0.01 to 0.015 millimeters in thin skin. Epidermal ridges are seen in the thick skin while they are absent in the thin skin. Hair follicles are absent in thick skin, they are present in thin skin. Erector pili muscle are absent in thick skin, they are seen in the thin skin. Sebaceous glands are absent in thick skin and they are present in thin skin. Sweat glands are numerous in the thick skin and they are few in the thin skin. Sensory receptors or Merkel cells are more in the thick skin, they are very few or less in the thin skin. Location: thick skin is seen in the skin of palm and sole. Thin skin is skin seen all over the body surface except the palm and the sole. Now that we have seen the differences between thick skin and thin skin, let us now see a diagram of thin skin. What we see here is the epidermal layer, the different stratum which we see here. This is the stratum basale, which is the deepest layer of epidermis. Then we see the stratum spinosum, 4 to 5 layers of polygonal cells or prickle cells. Next we see the stratum spinosum. What we don't see here is the stratum lucidum, which is absent. And next we see the superficial most layer, which is the stratum corneum, which is much thinner as compared to that seen in the thick skin. What we also see here is that the dermal papillae and the epidermal ridges are not that well seen here. What we also see here is the hair follicle, the sebaceous gland, and the erectoris pili muscle. We also see the sweat glands here, and we also see the dermis with connective tissue, blood vessels, and adipose sites. Now let us just quickly see the differences between the diagram of thick skin and thin skin. So we appreciate here that the stratum corneum is much thicker in thick skin as compared to that seen in the thin skin. Stratum lucidum present in thick skin, it's absent in thin skin. The dermal papillae and epidermal ridges are well differentiated and present in thick skin, while they are not that well appreciated in thin skin. The hair follicle with the sebaceous gland and the erectoris pili muscle present in the thin skin, and they are absent in the thick skin. Now we see a slide of thick skin, a low power view where we appreciate the epidermis, the thick stratum corneum, and also the presence of dermal papillae and epidermal ridges. Also see the reticular part of the dermis. There are no hair follicles, no sebaceous glands, and no erectoris pili muscle. A hyper view of the same shows us the strata of epidermis, the basal layer, that is the stratum basale, then the stratum spinosum, the basophilic appearing stratum granulosum, a small stratum lucidum, and a thick stratum corneum. We also have another clearer picture. This is a picture taken with the help of a software, that is the Apereo Image Scope software. We see here the different layers of epidermis. Clearly appreciate the dermal papillae and the epidermal ridges. A hyper view of this helps us in having a good view of the stratum basale, the stratum spinosum, the basophilic stratum granulosum, stratum lucidum, and a thick layer of stratum corneum. The salient features for spotting for a slide of thick skin would be presence of thick epidermis with stratified squamous keratinized epithelium, presence of prominent dermal papillae and epidermal ridges, and absence of hair follicles. Let us now see a low power field view of a slide of thin skin. What we see here is the epidermis, which is much thinner as compared to that seen in the thick skin. What we appreciate here is presence of hair follicles, which is the identifying feature here. A hyper field view of the same shows us the epidermis, not well differentiated dermal papillae and epidermal ridges, the dermis, and the presence of the hair follicles. Let us see a picture taken with the same software, giving a clear view of the epidermal layers and also presence of the hair follicles. Also appreciate the presence of the sebaceous glands and also the erectoris pili muscle, which is seen here in a higher magnification. Another higher magnification shows us the various components of the hair follicle clearly seen in this image. The salient features for spotting in a slide of thin skin will be presence of thin epidermis with stratified squamous keratinized epithelium, presence of hair follicles with sebaceous glands and erector pili muscles, and presence of unmarked dermal papillae and epidermal ridges. Thus, we have studied the two main slides of thick skin and thin skin in this video of Integumentary System Part 1. In the next video, we will be studying the cells of the epidermis along with the appendages of skin, that is the hair follicles, the sweat gland, the sebaceous gland, the erector pili muscle, and also the nail. Sontakke Yogesh, Textbook of Human Histology, CBS Publishers and Distributors Private Limited, New Delhi, 2020, page numbers 150 to 164. Thank you.